Hello friends, in this lecture we'll talk about the nucleic acids. Here are some previous year questions on the nucleic acids. Number one, which one of the following chemicals is a DNA intercalator? And the list is given, and among this list we have to select one molecule which acts as an intercalator. The another question was based on the number of or the different types of RNA molecules and their functions. These are listed in the two columns and we have to appropriately identify the correct combinations given in the different choices. Another question was based on the Chargaff's rule, where one DNA is mentioned and its cytosine composition is given. We have to identify or we have to predict how much the amount of A and T is present in that DNA. So let us have a look at the basics of the nucleic acids. So nucleic acids are the polymers of the nucleotides linked to each other by phosphodiester bond. These include deoxyribonucleic acid and ribonucleic acid, DNA or RNA respectively. Nucleotides are the building blocks of the nucleic acids, and the amino acid sequence of every protein in the cell and the nucleotide sequence of every RNA is specified by a nucleotide sequence in the cell's DNA. If we talk about the gene is the segment of a DNA molecule which contains the information required for the synthesis of a functional product, either DNA, either protein or an RNA molecule. The functions of these nucleic acids are different. For example, DNA has a function to store and transmit the biological information, whereas RNAs, they have broader functions, and several classes of RNA molecules are found in cells. Some of them are listed in this slide: ribosomal RNA, messenger RNA, transfer RNA, small nuclear RNA, miRNA, siRNA, and so on. So the nucleotides, they have three characteristic components. Number one, a nitrogenous base, a pentose sugar, and a phosphate group. So the molecule which lacks the phosphate group is referred as nucleoside. The nitrogenous bases are the derivatives of the two parent compounds, pyrimidine and purine, and accordingly they are referred as purine or pyrimidine nucleotides. So the parent molecules are shown over here. The number of the nitrogen or the carbon atom is also shown. So purines, they have two rings, and pyrimidines nucleotides, they have a single aromatic ring in their structure in the nitrogenous base. In sugar moiety, the carbon number is designated with prime symbol to distinguish the carbon number from the carbon of the nitrogenous base. So here the ribose sugar has a hydroxyl group on the 2' carbon of the pentose sugar, and the deoxyribose sugar is lacking a hydroxyl group on the 2' carbon, and because of this the RNA molecules are more reactive in nature. The phosphate can be attached to the 5' carbon or even at the 3' carbon, but the 5' nucleotides, they predominate, and in the nucleotides in the nucleic acid chain, the 5' nucleotides, they are present. The purines and pyrimidines are listed in this table. Purines include adenine and guanine, whereas pyrimidines include cytosine, thymine, and uracil. So adenine and guanine ribonucleotide or deoxyribonucleotide, they are found in RNA or DNA respectively. Guanylate is the ribonucleotide found in RNA, whereas deoxyguanylate is found in the DNA. Similarly, citidylate is the pyrimidine ribonucleotide which is found in RNA. Deoxycitidylate is found in DNA. Thymidylate is a ribonucleotide found in RNA. In the DNA, thymidylate is a ribonucleotide found in the RNA, whereas in DNA, deoxythymidylate is found. So thymidylate is often found in some RNA molecules such as tRNA molecule, transfer RNA molecule. Uridylate is exclusively found in the RNA molecule, which is a ribonucleotide. These are the structures of the nucleotides. Deoxynucleotides are shown, and you can see in case of purines, the nitrogen in the first position is linked to the first carbon Nitrogen in the ninth position is linked to the first carbon of the sugar in case of purines. And in case of pyrimidines, the nitrogen in the first position is linked to the first carbon of the ribose sugar or deoxyribose sugar in this case. In case of ribonucleotides also, the same kind of glycosidic bond pattern is seen among the purine and pyrimidine ribonucleotides. So these are the ribonucleotides shown over here: adenosine, guanosine, uridine, and cytidine. Okay. So here the basic difference between the uridine and cytidine is that there is an amino group in cytidine, whereas there is an oxo group. Two oxo groups are found in uridine. ATP is a nucleotide, and here two forms of the adenine nucleotide is shown, one in diphosphate form and another is in triphosphate form. ADP, when it gets phosphorylated, it gets converted into triphosphate form, ATP, which is used as an energy currency by the cell. Now these nucleotides, they are linked to each other by phosphodiester bond, where the 3' hydroxyl group of one nucleotide is linked to the 5' phosphate of another nucleotide. So likewise, several phosphodiester bonds are involved in linking the nucleotides, resulting in formation of a polynucleotide chain called as nucleic acid. So the DNA chain is shown, and the RNA chain is shown in this figure. Both have two terminal ends, where at one terminal end a phosphate group is free, and on another terminal end a hydroxyl group is free, and accordingly they are referred as 5' and 3' end respectively. So that gives the orientation to the nucleic acid chain. The nucleic acid sequence can be mentioned in this format, CAG, okay, or the orientation of the nucleic acid chain can be mentioned, writing with 5' and writing then followed by the nucleotide sequence, and then ending the sequence with the 3'. So if 5' to 3' is not mentioned, it is understood that the sequence is mentioned in 5' to 3' orientation. But for mentioning the sequence in 3' to 5', the always orientation must be mentioned. DNA structure. The native DNA is a double helix of the complementary antiparallel strands. In 1953, James Watson and Francis Crick proposed that DNA has a double helical structure. Their proposal was based on the extensive analysis of the X-ray diffraction patterns coupled with careful model building. DNA consists of two associated polynucleotide strands which wind together to form a double helix. The two sugar phosphate backbones are oriented on the outside of the double helix, whereas the bases are projected towards the interior of the helix. The adjoining bases in each strand, they stack on top of one another in parallel planes, and the orientation of the two strands is anti-parallel, which means that the 5' to 3' directions are opposite to each other. The strands are held in precise register by formation of base pairs between the two strands, where adenine always base pairs with the thymine and through double hydrogen bonds, whereas G, that is guanine, it always base pairs with cytosine through triple hydrogen bonds. So these base pair complementarity is the sequence, is the result of the size, shape, and chemical composition of the bases. Structure of the bDNA, which is the form which is exclusively or mostly found in the cells. So you can see over here the two strands, they run anti-parallel to each other, and the nitrogenous bases are oriented on the inner side, which form the hydrogen bonds with each other. So complementary base pairing is involved in this case, and the AT base pairs are linked through double hydrogen bonds, whereas GC base pairs, they are linked to each other by triple hydrogen bonds. Sugar phosphate backbone is oriented outward, and the two strands, they undergo twisting, resulting in helical structure formation, where two grooves are formed, major groove and minor groove. Now these grooves are the sites at which the DNA binding proteins can gain access through the nitrogenous bases present in the nucleic acid chain. Sharkaf's rule. Arvind Sharkaf proposed two rules for DNA based composition. Number one, in any double-stranded DNA, the number of guanine equals the number of cytosine residues and the number of adenine equals the number of thymine residues. Number two, the composition of DNA varies from one species to another. So accordingly, the molar ratio of A to T equals to 1. Similarly, the molar ratio of G to C equals to 1. The sum of the purines, adenine and guanine, always equals that of pyrimidines, that is cytosine and thymine. The percentage of C plus G does not necessarily equal the percentage of A plus T. So these are the things that you need to keep in mind. Different structures, different forms of DNA are found. Number one, bDNA, which is the most form of the DNA which exists in the right-handed helical structure. The extra diffraction pattern of the DNA indicates that the stacked bases are regularly spaced 0.36 nanometer apart along the helix axis. The helix makes a complete turn every 3.6 nanometer. Thus, there are about 10.5 base pair per helical turn, and this is referred as B form of DNA, which is the normal form of DNA present in most of the DNA stretches in these cells. In addition to this, the other forms are also found. For example, the A form of DNA, which is seen under low humidity conditions, and the RNA DNA duplexes as well as RNA helices, they take up the A form of DNA inside the cells or in vitro conditions. zDNA is another form of DNA where the short DNA molecules composed of alternating purine pyrimidine nucleotides, especially G's and C's, they adopt an alternative left-handed configuration instead of the normal right-handed helix. So this structure is called as zDNA, and because it appears, that molecule appears zigzag from the side. Some evidence suggests that zDNA may occur in cells, although its function is unknown. So there is the comparison of bDNA, aDNA, and zDNA. You can see in one complete helical turn, okay, the number of bases that can be accommodated in aDNA is slightly more. It is, for example, in aDNA, 11 base pairs per turn can be accommodated, whereas in bDNA, about 10.5 base pairs per turn can be accommodated. zDNA is the zigzag DNA that is shown in this figure. Finally, a triple-stranded DNA structure is formed when synthetic polymers of poly A and polydeoxyuridine are mixed in the test tube, and this can result in formation of a homopolymeric stretches of DNA composed of C and T residues in one strand and A and G residues in the another. These can form a triple-stranded structure by binding matching lengths of synthetic poly-CT. So such structures generally do not occur naturally in the cells. How the double-stranded DNA helix is stabilized? The stabilization occurs through non-covalent interactions between the stacking bases. So stacking interactions they contribute majorly for the stabilization of the helix. So stacking interactions between the adjacent bases and the hydrogen bonding between the complementary strands, they contribute to the stable structure of the DNA helix. So among the stacking interactions, the hydrophobic and van der Waals forces, they contribute to the stabilization of double helical structure, whereas the presence of several hydrogen bonds, thousands of hydrogen bonds in a DNA molecule contributes greatly to the stability of the double helix. Now DNA has a property of undergoing reversible strand separation. So if the DNA solution is subjected to high temperature, then the melting of the DNA occurs, which occurs due to strand separation. So strand separation can be referred as denaturation or melting. So here, as the strands they get separated, the absorbance of the DNA solution increases. Here the absorbance of the nucleic acid single-stranded DNA is shown, where as the temperature increases, absorbance also increases. But you can compare it with the double-stranded DNA. It is more in case of single-stranded DNA. And the TM value is also affected by the base composition. For example, the GC, especially the GC base pairs, they contribute to the TM value. They contribute more to the TM value because as they are linked to each other by triple hydrogen bonds, the more energy is required to break such bonds. And so if the DNA has more GC content, its TM value will be more. So as GC content increases in the DNA, the TM value of those DNA molecules also increase. The renaturation conditions. The denatured DNA can undergo renaturation, and this can be carried out by bringing the conditions to the normal. For example, lowering the temperature or increasing the ion concentration or neutralizing the pH can result in renaturation of the DNA strands. The extent of such renaturation is dependent on time, DNA concentration, and the ionic strength. Many DNA molecules, they occur in a circular manner. For example, prokaryotes, they have circular chromosomes. Many viral genomes which contain DNA, they have circular genomes. Mitochondria also has a circular DNA molecule. These DNA molecules, they can undergo underwinding or overwinding. Underwinding or overwinding can result in supercoiling. So if the DNA is underwound, negative supercoils will be there, and if the DNA is overwound, then it results in positive supercoils. Now these supercoils, they can be removed by the action of topoisomerases like topoisomerase 1 and topoisomerase 2. This is the structure of the DNA molecule, which is having two topological forms. One is in the compact form, supercoiled form, and another is in relaxed form. Relaxation occurs due to breakage in the one of the strand of the double-stranded DNA. So the same DNA molecule can get converted into the relaxed configuration. The E. coli nucleoid is shown over here, which is highly compact, and you can see several supercoiled regions are found in the DNA. Now here the mobility of these DNA molecules in different topological forms, they can differ from each other. So here the relaxed DNA and supercoiled DNA is shown. When such DNA solution is subjected to electrophoresis, two separate bands of the DNA are observed, where the compact DNA, which is the supercoiled DNA, which it will travel the most distance from the loading site, and whereas over here the nick circular DNA molecule or open circles, it will travel a less distance because the compaction is lost because of this nick. The intercalating agents such as ethidium bromide can either introduce or remove the supercoils. So for example, over here negatively supercoiled DNA is shown. If it is mixed with ethidium bromide at some concentration, it will result in relaxation of the supercoiled DNA, and further increase in the ethidium bromide can result in addition of the supercoils. So this ethidium bromide, it gets intercalated between the adjacent base pairs. Similar to ethidium bromide is an acridine orange. Acridine orange is also an intercalating agent. RNA molecules, the primary structure of RNA is generally similar to that of DNA with two exceptions. Number one, the sugar component of RNA is ribose, which has a hydroxyl group at the 2' position, and thymine in DNA is replaced by uracil in RNA. The hydroxyl group on C2 of ribose sugar makes the RNA more chemically labile than DNA and provides a chemically reactive group that participates in several RNA-mediated catalysis. Primary and secondary structure of RNA. Unlike DNA, which exists primarily as a very long double helix, Most cellular RNAs are single-stranded and exhibit a variety of conformation. Differences in the sizes and conformations of the various types of RNA permit them to carry out specific functions in the cell. So several secondary structures are found. These are listed: hairpin structure, stem and loop structure, pseudoknot structure. So hairpin structures are formed by pairing of bases which are separated from each other by just 5 to 10 nucleotides. Whereas stem and loop structures are formed by pairing of bases that are separated from each other by more than 10 nucleotides to several hundreds of nucleotides. Pseudonodes are formed when simple folds cooperate to form more complicated tertiary structure. So here is the figure in which the hairpin structure is formed where two complementary regions are separated from each other by 5 to 10 nucleotides. If the complementary regions are separated by 10 to hundreds of nucleotides, then stem and loop structures are formed. In case of pseudonodes, the two stem and loop structures, they interact with each other where stem region of one stem portion of one stem and loop structure becomes part of the loop structure of another stem and loop region. So such structures results in further folding of the RNA molecule. Single-stranded RNA exists in the helical form, and internal base pairing also can result in further changes in the conformation. Types of RNA molecules. Different types of RNA molecules are found. The messenger RNA molecule. Messenger RNA molecule is the template for protein synthesis on ribosomes, and messenger RNA molecule may be produced for each gene or group of genes that is to be expressed in E. coli, whereas a distinct messenger RNA is produced for each gene in eukaryotes. Messenger RNA is a heterogeneous class of molecules. In E. coli, average length of an mRNA molecule is about 1.2 kilobases. Transfer RNA is another class. It carries the amino acid in the activated form to the ribosome for peptide bond formation in a sequence that dictated by the messenger RNA template. So it acts as an adapter molecule. So for each kind of amino acid, there is at least one kind of tRNA molecule. tRNA molecules consist of about 75 nucleotides to 90 nucleotides length, and this makes the tRNA molecules smallest among the RNA molecules. Modified bases are often found in the tRNA molecules such as uridine, pseudouridine, and dihydrouridine. So these are the modified bases that are found in the tRNA molecule. You can see the structure of the tRNA molecules. At the 3' end, there is a CCA residue, which is the site of the amino acid attachment. Then T-Cy-C loop is there. Thymine is present in this loop region. Variable loop has a dihydrouridine. D loop also has a dihydrouridine, and this is the anticodon loop, which is used to identify or recognize the codon on the messenger RNA on the ribosome. Ribosomal RNA, these are the major component of the ribosomes, which play both a catalytic and a structural role in protein synthesis. In E. coli, there are three kinds of ribosomal RNA: 23S ribosomal RNA, 16S, and 5S ribosomal RNA. Because of their sedimentation behavior, they are named differently. One molecule of each of these species of ribosomal RNA is present in each ribosome. So here 70S ribosome found in the bacteria and 80S ribosome found in eukaryotes. They are made up of ribosomal RNA and proteins. So 70S is composed of 50S subunit and 30S subunit. 50S consists of 5S ribosomal RNA and 23S ribosomal RNA, whereas 16S ribosomal RNA is found in the small subunit of the ribosome. Similarly, the large subunit of the eukaryotic ribosome consists of 5S ribosomal RNA, 28S ribosomal RNA, and 5.8S ribosomal RNA, whereas the small subunit is composed of 18S ribosomal RNA in case of eukaryotes. The internal base pairing also results in further folding of the RNA molecules. You can see different conformations of the 16S ribosomal RNA and 5S ribosomal RNA are shown. Ribosomal RNA is the most abundant of the three types of RNA. Transfer RNA comes next, which is followed by the messenger RNA, which constitutes only about 5% of the total RNA. Other RNA molecules include small nuclear RNA molecules, snRNA, which participate in the splicing of the RNA exons. They are components of the spliceosome complex. SNORNA are the small nuclear RNA, which is a class of small RNA molecules that are involved in chemical modifications of ribosomal RNAs and other RNAs, for example, methylation. SNORNAs are component of small nuclear ribonucleoprotein, SNORNP, which contains SNORNA, RNA and proteins. siRNA and microRNA, these are some small RNA molecules which cause RNA interference, and which is a mechanism for gene regulation in all eukaryotes. siRNA and miRNA are the central to RNA interference. So siRNA or small interfering RNA molecules, they are approximately 21 nucleotides long, and these molecules silence the gene expression by binding to the target mRNA in association with the RISC complex and causes degradation of the target mRNA. Whereas microRNA are small non-coding RNA molecules encoded in the genomes of plants, animals, and their viruses. Approximately they are 20 to 25 nucleotides long, and they cause gene silencing by binding to the target mRNA in association with RISC complex and thereby blocking the translation of messenger RNA. So this is the basic difference between siRNA and miRNA. So let us have a look at the questions. Which one of the following chemical is a DNA intercalator? 5-bromouracil, ethyl methane sulfonate, acridine orange, or UV. Now you can see all these are mutagens, but what is asked, which one of these is an intercalator molecule? So 5-bromouracil and ethyl methane sulfonate, these are the chemical mutagens. These can be base analogs, whereas UV is a physical mutagen. Acridine orange is an intercalator and it gets inserted between the adjacent bases and cause insertion mutations. So acridine orange is the correct answer. Next one, here the list is shown about the different RNA molecules, and you have to find out the functions of the RNA molecules. So snRNA molecules, what are the functions of the snRNA, siRNA, microRNA, snRNA? So just whatever the RNA molecule whose function is absolutely known to you, just try to identify such kind of combination. So siRNA is the one in which they turn off the gene expression by directing degradation of selective messenger RNA, and miRNA, they block. Okay, so they block the translation of the selective messenger RNA. Okay, so this is the one. So you just identify B1 and C2. B1 and C2 are there. So these are the two options in which B1 and C2 are there. So then try to find out the other one. So snRNA, its function is in a variety of processes including splicing and pre-messenger RNA. So this is the appropriate one. So the most appropriate answer is the second one. Another question is there, which is based on Chargaff's, where C is found to be 20%, and you have to find out the A and T put together. So here C is 20%. So similarly, G would be 20%. 20 plus 20 will be 40%, and the A and T put together will contribute to 60%. So 60% is the correct answer, which is based on the Chargaff's rule. So this was about the nucleic acids. The major reference is Leninger's Principles of Biochemistry and Molecular Biology of Gene by J.D. Watson. Thank you and happy learning.